Hi Hires, so as well as giving you access to this PowerPoint just as is and giving you as a PDF printout so that you can add your own notes to it, I'm also going to do narrations for most of the PowerPoints which you can use and go through taking notes as you go, or just so that you've gone through it with me, as it were. Okay, so we're looking now at nucleic acids, and you've actually been told about them, but in pretty broad stroke detail. Now, so for a start, like, why are they called nucleic acids? So nucleic acids is a name that we're kind of left over with because in biology this happens quite often. We find things and you get named because of where they found them or what they looked like. So nucleic acids were originally found out from the nucleus. So they were smashing up cells and separating them out in different ways, using centrifuges, using filters, and then when they got into the bits of the cells, they were then separating out the chemicals, and they found these big, long-stranded chemicals. And they tested them the normal way you would test things. So they test for, are they soluble? Do they react with acids, alkalis, ethanol, things like that. So they were looking for chemical properties, and the simplest one they found was they were all acid. So they said, well, these are acidic, so acids. They found them from the nucleus, so they said, well, that's nucleic. So they called it nucleic acids. And every single one of these, they then found out, were made up of subunits. So if they broke it down even further, each one was made up of something called a nucleotide, and you're going to need to know it in quite a lot of detail. Okay, so there are some basic nucleic acids. You already know DNA, deoxyribonucleic acid. You're going to know it in a lot more detail later. And you should have heard of RNA, which is ribonucleic acid. The only RNA you've actually heard of is this one, which is messenger RNA, which you should know takes a copy of your DNA code out from the nucleus to the ribosome, okay, where it's then used as the basis to code for your proteins. These ones you haven't heard of yet. tRNA, which is transfer RNA, and rRNA, which is quite difficult to say, ribosomal RNA. You do have to write it like this, by the way. You have to have a small letter followed by your capitals. So you know what the ribosome is for? You know, the ribosome is to make proteins. So ribosomal RNA must be part of the ribosome and involved with making proteins. In fact, both of these two are. They are involved in something called translation, which is where you translate your DNA code into a proper protein. Actually, you translate your mRNA, but that came from your DNA. Right. So a nucleotide. So a nucleotide is the subunit, and the subunit is the same basic structure in all nucleic acids. Okay, so it starts off with a phosphate. Now you've heard of phosphate, but you probably heard of it in this. Does that sound familiar? Adenosine triphosphate. If you are a chemist, you've done some chemistry, you might have heard of phosphate already. I'm going to show you what it looks like properly in the next slide. And the phosphate is attached to a sugar. Now the sugar is what is different to make DNA, DNA, and RNA, RNA. And finally, we have a base. And the base, you should actually know something about this. In fact, if you want to, do a pause and see if you can remember, write down the names of the bases that you think there should be in nucleic acids. Wee pause symbol. Okay, so a phosphate. I said I'd give you the real shape of a phosphate. Phosphate is actually this formula. So I was saying if you were a chemist, you might have seen it. If you're doing anything to do with complex ions, this is in the complex ions page. This is what it looks like. So it's one of these very small molecules. You can see why actually you're quite glad that we're just going to draw it always as a little circle. This packs in quite a big punch for a molecule. It's got 3 negative charges. This is big and heavy hitting. That negative charge will decide things like how DNA runs in a gel. We're going to talk about that later, but that's like how you do DNA fingerprinting. It's how you separate out DNA for genetic engineering. It's really, really important, and this is what allows that to happen. It's also used not in DNA, but using it from ATP to activate proteins, deactivate proteins, because it does just do lots because it's got this big, big, big negative charge. Okay, right, we're ready for the sugar, because the sugar is actually quite complicated. You need to know some very precise things about it. So the sugar is actually different in DNA and RNA. In RNA, it's ribose, so ribonucleic acid, and deoxyribose, deoxyribonucleic acid, in DNA. Okay, so they are very similar sugars in terms of size. They have both got 5 carbons. And so if I was going to draw ribose properly, this is what I draw. This is actually a carbon here, a carbon here, a carbon here, and a carbon here. So 1, 2, 3, 4, 5. Now that does look quite complicated. You are normally going to draw it as the pentagon that you can see there. Sorry, the pen is not the best for me to draw with. Okay, so you have a pentagon, which is this shape. Okay. Oh, that's gone a little bit wobbly. And also you have to draw this bit coming up here, because this is another carbon up here. So to show what's going on, you actually need to be able to number this shape. You start with the number 1 here. Okay, oh, that's how they join on. So 1, 2, 3, 4, 5. So this one up here, there would be 5. And that would join on to the phosphate of the one above. And this one joins on to the phosphate going down the way. Okay, so 1, 3, and 5 are the numbers that you need to know, and you need to know where they are on that shape. Okay, right, here's deoxyribose. Okay, you can do another little pause and see if you can spot the difference. You got it? Difference is here. I have taken away this oxygen. So if you are dehydrated, you've lost water, you don't have any water present. So if I have deoxide, I have taken off the oxygen. You don't need to know that detail. I'm just saying this is what that is. Okay, it has exactly the same overall shape though. If I kind of put this here, here's my 1, 2, 3, 4, 5 up here. That will attach onto the phosphate, and this one will attach onto the phosphate going on to the next one. Okay, this one here will attach onto the base in exactly the same way as that one did. Okay, so this is what you have to be able to draw. These kind of slightly neater than the ones I've sketched. Not all of the H's and OH's, but the kind of basic pentagon shape. So can't read that behind it. So the carbon numbers are really important because they give what's called a direction to the chain. So if we were going to actually look at these properly, the real bases, I am not going to really go into detail in these, but there are actually 5. I asked you to note down which ones you think you could remember. I'm expecting or hoping that you managed to get adenine, which is A. You get that in both of them. Thymine, T, A to T. Remember that as a complementary base pair. G, guanine, and C, cytosine. But there is a new one. So you'll notice that thymine, you only get that in DNA, but where it doesn't say and RNA. So we need something else that's going to match up to A in RNA when you're making it. And for that you use something called uracil. So this bit, this is new. Need to learn that bit. Okay, so the pairing is the essential thing. So it goes A to T, and it also goes A to U, and G to C is always the same. You can totally ignore this bit because this is what they really look like, but frankly I'm happy with us just putting a little box and labeling it base, and that is everything. Okay, these are sometimes referred to as nitrogenous bases, and you can see why they're called nitrogenous, because they've got N's everywhere. And nitrogens, well, that's another reason. Nitrogen cycle, this is another reason why it's essential. If you don't have constant nitrogen availability, then you won't be able to make DNA or RNA, and that's, your cell will die. Just for curiosity's sake, you can have a look here. Can you see why the strands are always exactly the same distance apart? DNA has A to T and G to C. Now I'm going to tell you, just give you the hint. Let's do the number of rings. And I'm moving on. Okay, so I put all that together. and we get something that looks like this. But we're going to draw that as that. And you need to be able to do that numbering. So that's a 1, that's a 3, and there's the 5 in there. Okay, so it looks a lot less stressful than this, but you have to not be thrown by them showing you something that complicated. Okay, and your numbers as you're going through. Okay, so the carbon number, as I say, that's really important to give you the direction of the chain. This one is called the 5' end, and it's quite, and normally we put a little tag on it just to show this is the 1' carbon, 2' carbon, 3' carbon, 4' carbon, and 5' carbon going around. Okay, so the 5' attaches to the phosphate, and the 3' is going to attach to the next phosphate. Okay, so the next nucleotide. And then what we end up with is the sugar phosphate backbone, and you can see here's your 5 to 3, 5 to 3, 5 to 3, 5 to 3. So this end, at this top bit, there's always going to be a 5' carbon, and at the bottom there's always going to be a 3' carbon. So this is what's called the direction of the chain, and it matters for stuff like replication. Okay, so this is a chain linking together 5' to 3'. Every single one of these bonds is strong. They are covalent bonds. It is not easy to break these, which is good because you don't actually want to. So the bases are actually going across the way with complementary pairing, and that's not changed since Nat5. So your A to T, your G to C. Okay, but they're going to link together with hydrogen bonds. So these bonds in between here, they are strong enough, they'll hold the DNA together, but they are much weaker than the covalent bonds. So what that means is if you want to pop open a bit of your DNA so that you can like copy out a code, you only have to snap the weaker bonds. So it's easy to do that, and as you're pulling at it, the rest of the chain doesn't just dissolve. So really, really important that these are weaker and that they do the pairing the right way. So what the bases give you is the double strand of DNA. It also gives you RNA, but we're not going to talk about that just quite yet. Okay, so there's a problem with this though, because that picture of a nucleotide, this kind of thing here, you got to remember this is actually three-dimensional. This has very, very precise angles going on here. Carbon to carbons are 109.5 degrees. But you have a problem with this because if I have my normal kind of branching thing going down here, sorry, my chain, if I try and match up going this way to another base, it doesn't fit. I have to flip over the other strands. So what I end up with is anti-parallel strands. So what we mean by anti-parallel is, well, here's a 5 up here, there's your 1, 2, 3, oh, sorry, 1, 2, 3, 4, 5. So that's the 5, there's your 3, there's the 5, there's your 3, all the way down to, so that's the one that we had set up before. So this is kind of the normal strand, that's fine. But this strand, when I try and put a second one on, I can't do it this way around. So what I need to do is flip it upside down, as it were. So now I've got a 5 here. So there's my 1, 2, 3, 4, 5. So there's my 3, there's my 5, there's my 3, there's my 5, there's my 3, there's my 5, and I'm now at my 3. So this is the 3' end, here's your 5, here is a 3, here is a 5. You have anti-parallel strands. So you now have how you are going to talk about your DNA. Instead of you saying it's a double-stranded double helix, which is what you could say at the end of 4th year, you're now going to say it is a double-stranded double helix with anti-parallel strands. So the best way, I think, to get that, if you think of a ladder and then you basically twist that, hold one bit at the top, twist it from the bottom, this is what you would get, is that's your double helix. So the whole point of it, like why have we got it? It is because this base sequence that we get on the DNA forms everything about what the cell needs to be able to do. We kind of skimmed over quite a lot of it. We talked about just saying your DNA codes for specific characteristics, each of those codes being a gene. But there's a lot more to it than that. Your DNA decides things like exactly how much of a protein you'll make, when you'll make it, exactly how the cell's going to behave. It's going to do a huge amount of quite complicated things, because it is not just the information in the cell, it is then everything to do about controlling the cell. And you did say that even in second year, you said the nucleus controls the cell, but you're going to have to actually be able to explain how it controls the cell. So I think this stuff is super fascinating. You just have to go and learn the specifics on a nucleotide.